Welcome to the deep dive, where we cut through the clutter and really get into the fascinating stuff. Today, we're taking a journey deep inside your body's incredible control systems.
That's right.
If you're maybe just starting out in medicine, think of this as your roadmap. We're focusing on how everything stays balanced, especially the endocrine system and, well, the pancreas. It's a real powerhouse.
Absolutely essential stuff.
Our mission today, to really understand how your body maintains that perfect internal balance. You know, what we call homeostasis.
Mm-hmm.
And it does this through this complex network of chemical messengers. So we'll break down the control systems, get familiar with those messengers' hormones, and then really zoom in on insulin and glucagon from the pancreas.
Two key players, definitely.
Just think about it for a moment.
Yeah.
How does your body know when to sweat or when to store energy versus releasing it?
Yeah, it's happening constantly.
All automatically, right? Beneath your conscious radar, we're going to try and uncover some of that elegant design behind the regulation.
It's a beautiful system.
So let's start with a core idea. Imagine your body is like the smartest smart home ever.
Okay.
It needs a central heating and cooling system, constantly adjusting things, right?
Yeah.
To keep the temperature just perfect.
Exactly. That's a great analogy. That's basically what a control system does in the body.
Okay, so its job is homeostasis.
Precisely. Maintaining that stable internal environment, no matter what's happening outside. It's always working to keep key things, like blood glucose or body temperature, right at their optimal set point.
It's pretty amazing. It works so well.
It really is.
And you mentioned every good control system needs key parts. I like the anchor RCCE for this.
Yes, that's a great way to remember it. Receptor, control center, and effector. RCCE.
Right, so the receptor, that's the sensor.
Exactly. The receptor is the body's lookout, its watchman, constantly monitoring the variable it's responsible for, like thermoreceptors in your skin checking the temperature.
Gotcha. Then that info goes to the control center.
Yep.
That's the brain of the operation. It compares the real-time value from the receptor to the ideal set point. Often, that's the hypothalamus in the brain for endocrine stuff, or maybe the medulla oblongata for breathing.
And it decides if something needs to change.
It does, and then sends instructions to the effector.
The action hero.
Yes. The effector is the component that actually makes the change happen, brings the variable back in line. So if you're too hot, your sweat glands are the effectors. They get the signal and start cooling you down.
A coordinated team effort.
Absolutely. And communication between them is obviously key.
Right, they need to talk to each other constantly. How does that happen?
Well, often through what we call feedback loops. Think of it as the body's regulatory dance. The most common type, the one that keeps things stable, is negative feedback.
Ah, the thermostat example again. Temperature drops, heater comes on, temperature hits the target.
Heater turns off. That turning off is the crucial part of negative feedback.
In the body, it works to reduce or reverse the original stimulus. So take that fever example. Thermoreceptors sense high temp, signal the hypothalamus. Hypothalamus activates sweat glands, the effectors.
You sweat, you cool down.
And that cooling reduces the high temperature, which then tells the hypothalamus to stop activating the sweat glands. The system switches off once you're back near the set point.
So it prevents things from going too far in one direction.
Exactly. It maintains stability within tight limits.
Okay, so here's a question for you listening. Thinking about that switching off mechanism, how might that apply to, say, your blood sugar levels after you eat a big meal? Keep that in mind. We'll definitely come back to it when we discuss insulin.
Good question.
Now, the other type, positive feedback. You said this one's rarer and maybe more dramatic.
Yes, much rarer for everyday homeostasis. Instead of turning off the response, it amplifies it. It ramps things up, like hitting the gas pedal.
So not great for keeping things stable day to day.
Generally, no. It can lead to runaway processes if not carefully controlled, but it's essential for specific short-term events. Think blood clotting.
Oh, right.
When you get a cut, chemicals are released that attract platelets. Those platelets then release more chemicals, attracting even more platelets, snowballs, amplifies rapidly until the clot forms and seals the wound.
And other examples?
Ovulation, the release of an egg, involves a positive feedback loop. And childbirth, the contractions get stronger and stronger due to positive feedback. It serves a specific, intense purpose.
Sort of like a controlled explosion almost.
In a way, yes, a very targeted amplification for a specific goal. Which leads to another question for you. Given that amplifying nature, why do you think positive feedback loops are kind of reserved for these specific often temporary events, rather than, the constant adjustments needed for daily balance, something to ponder.
Interesting. And you also mentioned the set point itself isn't always rigid. It can change.
That's right. It often varies over a 24-hour cycle. This is your circadian rhythm, your body's internal clock, deeply influenced by light and dark cycles.
Like jet lag.
Exactly. That feeling is your internal clock being out of sync with the external environment. Think about cortisol, the stress hormone. Its levels naturally peak around 7 a.m., helping you wake up.
And then they drop in the evening.
Precisely. They dip around 7 p.m., preparing you for sleep. Melatonin, on the other hand, released by the pineal gland, rises in darkness to promote sleep.
So our bodies have these daily rhythms built in.
They do,
Yeah.
guided by a tiny part of the hypothalamus called the suprachiasmatic nuclei, or SCN. It takes cues from the environment. We call these Zeitgebers, German for time givers. Light is the most powerful Zeitgeber.
Fascinating. So we have control systems, feedback loops, rhythms. But how are the messages actually sent? We mentioned chemical messengers.
Yes, hormones. These are the body's chemical signals, like tiny targeted text messages. They're produced in specialized endocrine glands, released into the bloodstream, and travel to affect specific target tissues elsewhere in the body.
And they travel through the blood.
Mostly, yes. That's the endocrine effect, traveling far. But they can also act locally on neighboring cells, that's paracrine, or even act back on the cell that made them, which is autocrine.
They move fast.
Surprisingly fast. A hormone molecule can circulate through your entire bloodstream in about 30 seconds.
Wow, okay. And where are these hormone factories, the endocrine glands?
They're distributed throughout the body. Up in your head and neck, you've got the pituitary, pineal, thyroid, parathyroids, hypothalamus.
Right.
In your abdomen, there are the adrenal glands sitting on top of the kidneys, the pancreas, even parts of the kidneys and gut make hormones. And then in the pelvis, the gonads, ovaries, and testes, and during pregnancy, the uterus and placenta.
So they're really everywhere.
Pretty much a whole network of chemical communication centers.
Now, to keep track of all these different hormone messengers, you mentioned a classification system, PAES, was it?
Yes, PEGs. It helps us understand how they're made, how they travel, how they act. It stands for peptide polypeptide, amino acid derivatives, glycoprotein, and steroid hormones.
Okay, let's break down PAGE, peptides first.
Right. Peptide polypeptide hormones. This is the biggest group. They're basically chains of amino acids. Crucially, our stars today, insulin and glucagon, belong here. Most peptides are hydrophilic.
Hydrophilic, water-loving.
Exactly. Think of them like open postcards. They dissolve easily in the watery blood plasma and travel freely.
Okay, next in PAGE is A.
Amino acid derivatives. These are modified from single amino acids, usually tyrosine. Examples include adrenaline, your fight or flight hormone, which is hydrophilic, but also the thyroid hormones, T3 and T4, which are actually hydrophobic.
Ah, okay, so some variety there. Then G.
Glycoprotein hormones. These are large proteins with sugar chains attached, like thyroid stimulating hormone, TSH from the pituitary. Also hydrophilic, like the peptides.
And finally S.
Steroid hormones. These are all derived from cholesterol, a lipid, so they are inherently hydrophobic, water-fearing.
Are lipid-loving VIPs needing a ride?
Exactly. Think cortisol, testosterone, estradiol. They don't mix well with water.
So that brings us back to your earlier question. Why does being hydrophilic or hydrophobic matter so much? How does it affect their travel and how they talk to cells?
It's absolutely crucial. Let's talk transport. The hydrophilic ones, the peptides, glycoproteins, adrenaline, they're fine on their own in the blood. They just dissolve.
The open postcards.
Right. But the hydrophobic ones, steroids, thyroid hormones, they can't just dissolve in watery blood. They need a chaperone.
The VIPs needing a ride.
Precisely. They bind to carrier proteins in the blood plasma. These proteins act like taxis, shielding them from the water and carrying them around.
So they're mostly bound up.
Yes. And here's the key thing. Only the tiny fraction that's unbound or free is actually biologically active and can interact with cells. For thyroid hormones, it's less than 1% that's free and active.
Wow, okay. So that answers the transport part. Hydrophobic hormones need carrier proteins because blood is mostly water. How does it affect how they interact with cells?
Big difference there, too. Hydrophilic hormones, being water-soluble, generally can't cross the fatty cell membrane easily, so they bind to receptors on the surface of the target cell, like knocking on the door.
Okay.
But hydrophobic hormones, being lipid-soluble, can slip right through the cell membrane. They typically bind to receptors inside the cell, often directly influencing gene activity in the nucleus. They walk right into the cell's control room.
Very different mechanisms of action then.
Fundamentally different.
So the body makes these hormones.
Mm-hmm.
But how does it know when to release them? You mentioned feedback loops earlier.
Yes, negative feedback is the main controller for most hormone secretion. And often it involves a cascade, where one hormone triggers the release of another.
Like a chain reaction.
Kind of. Many hormones are tropic hormones. Their job is to stimulate another endocrine gland to release its hormone. Think of the hypothalamic pituitary adrenal HPA axis. It's like the central command for stress.
Okay, how does that work?
The hypothalamus releases CRH. That tells the pituitary to release ACTH. ACTH then travels to the adrenal glands and tells them to release cortisol.
A three-step process.
Right. And then high levels of cortisol and, to some extent, ACTH feed back to the hypothalamus and pituitary and say, okay, enough CRH and ACTH for now. That's the negative feedback loop, shutting down the initial signal.
Keeping the stress response from going overboard.
Exactly. TSH stimulating the thyroid is another classic tropic hormone example.
All right, this is a great context. Now let's zoom in on our star organ for today. The pancreas. You said it's behind the stomach.
Yep, tucked away back there, about 15-20 centimeters long. And it's a real multitasker.
How so?
Well, biologically, it has two major parts. The vast majority, like 98%, is the exocrine pancreas. This part makes digestive enzymes amylase for carbs, lipase for fats, proteases for proteins that get secreted into the small intestine.
Essential for digestion.
Absolutely. But the other 2%, that tiny fraction, is the endocrine pancreas, and that's our focus.
That's where the hormone action is.
That's where it is. Within that 2% are these little clusters, like tiny islands of cells, called the islets of Langerhans.
The famous islets.
Famous for a reason. These islets contain different cell types, but the main ones we care about for blood sugar are the alpha cells and the beta cells.
Okay, what do they do?
Alpha cells, making up about 20% of the islet cells, produce glucagon. Beta cells, the majority, around 75%, produce insulin.
Insulin and glucagon, the dynamic duo.
The absolute key players in blood glucose regulation. They work in opposition, like a seesaw, to keep your blood sugar levels incredibly stable.
And this directly answers that earlier question, right? How the body knows when to store or release energy based on blood sugar. It's these cells sensing it.
Precisely. And it connects back to negative feedback. Remember we asked how blood sugar control might work like a thermostat?
Yeah.
When your blood glucose rises after a meal, the beta cells sense this and release insulin. Insulin then acts to lower the glucose, as the glucose level comes back down towards the set point.
The beta cells stop releasing insulin.
Exactly. The drop in glucose inhibits further insulin release. That's the negative feedback. It switches off the response when the job is done.
Elegant. Let's dive deeper into insulin. You called it the storage manager.
That's a good way to think of it. Its main job when blood glucose is high is to promote energy storage, get that glucose out of the blood and into cells to be used or stored away.
And it was a peptide hormone, part of Pancreas, hasn't made.
It's a classic protein synthesis pathway. Starts as a large precursor, pre-proinsulin. A signal piece gets snipped off, leaving proinsulin.
Okay, like an intermediate step.
Right. Then proinsulin holds up, gets some structural bonds formed, and is sent to the Golgi apparatus where it's cleaved one last time into active insulin and another fragment called C-peptide.
C-peptide.
Yeah, it's released along with insulin in equal amounts. Clinically, we can measure C-peptide levels to get an idea of how much insulin the body is actually producing itself. Both insulin and C-peptide are then packaged into vesicles, ready for release.
So how does the beta cell get the signal to release these vesicles, glucose itself?
Glucose is the primary trigger. It enters the beta cell through a specific transporter called GLUT2.
Okay.
Inside the cell, glucose metabolism ramps up, producing ATP, the cell's energy currency. This rise in ATP causes specific potassium channels on the cell membrane to close.
Right.
Closing those channels changes the electrical charge across the membrane. It depolarizes. This depolarization triggers voltage-gated calcium channels to open. Calcium ions flood into the cell.
And calcium is the trigger.
Calcium is the key final signal. That influx of calcium tells those vesicles containing insulin and C-peptide to move to the cell membrane, fuse with it, and release their contents into the bloodstream. It's called exocytosis.
Wow, that's quite a cascade. From glucose coming in to insulin going out.
It's a beautiful, finely tuned mechanism. And interestingly, some drugs used for type 2 diabetes, like sulfonylureas, actually work by targeting those ATP-sensitive potassium channels to stimulate insulin release, even when glucose isn't super high.
Clever. Okay, so insulin is released. How does it act on its target cells, like muscle and fat?
Insulin binds to a very specific receptor on the surface of those cells. It's called a tyrosine kinase receptor. Think of it like a complex lock on the cell door.
Tyrosine kinase.
Yeah, it means when insulin, the key, binds to the outside part of the receptor, the inside part gets activated and starts adding phosphate groups, phosphorylating specific tyrosine amino acids on itself and other proteins inside the cell.
So binding triggers internal chemical changes.
Exactly. It sets off a signaling cascade inside the cell. This cascade ultimately leads to insulin's metabolic effects.
Which are mainly about storage, you said, anabolic effects.
Yes, insulin is fundamentally anabolic building up. It promotes glucose uptake by cells. It's glycogenic, stimulates the liver and muscles to store glucose as glycogen. It's anti-gluconeogenic. It tells the liver not to make new glucose from scratch. It's anti-lipolytic, stops the breakdown of stored fat, and it promotes protein synthesis.
So basically, it lowers blood glucose by pushing it into cells and storing it.
That's the bottom line, lowering blood glucose.
Okay, now for the other side of the coin, glucagon, the mobilization expert.
Perfect description. Glucagon steps up when blood glucose levels are low between meals, during fasting. Exercise, its job is to raise blood glucose.
Also a peptide hormone.
Yes. Also made in the pancreas, but in the alpha cells. It's also synthesized as a precursor and then cleaved to its active form, a single chain of 29 amino acids, ready to mobilize energy reserves.
And how does it signal? Does it use the same kind of receptor as insulin?
No, it uses a different type, a very common one called a G-protein coupled receptor, or GPCR. It binds to this receptor mainly on liver cells and, to some extent, fat cells.
Yeah, PCR. Okay.
Binding to the GPCR activates an enzyme inside the cell called adenylate cyclase. This enzyme makes a small molecule called AMP, cyclic AMP, which acts as a second messenger. AMP then activates another enzyme called protein kinase A, PKA.
Another cascade.
Another signaling cascade, yes. PKA then phosphorylates various target proteins, leading to glucagon's effects. Different pathway, different outcome compared to insulin.
And those effects are opposite to insulin's.
Yeah.
Catabolic.
Exactly. Glucagon is catabolic, breaking down stores. It's glycogenolytic. It triggers the breakdown of stored glycogen in the liver, releasing glucose into the blood. It's gluconeogenic. It stimulates the liver to make new glucose from amino acids and other precursors. Its lipolytic, promotes the breakdown of fat in adipose tissue. And it can be ketogenic, stimulates the liver to produce ketone bodies from fatty acids, another fuel source.
So its main job is raising blood glucose.
Absolutely. Raising blood glucose by releasing stored fuel.
So you really see them as this perfectly balanced pair, insulin and glucagon, constantly adjusting based on blood sugar levels.
It's a continuous, delicate balance. Insulin dominates after a meal when glucose is high. Glucagon dominates during fasting when glucose is low. Together, they keep blood glucose remarkably stable in healthy individuals.
Which is obviously crucial for health.
Yeah.
What happens when this balance is lost?
Well, that's where we get into clinical problems. If you have too much insulin action, either from too much injected insulin or maybe a rare insulin-producing tumor, you can get severe hypoglycemia, dangerously low blood sugar. Much more commonly, if you don't produce enough insulin, like in type 1 diabetes, or your cells don't respond properly to the insulin you do make that's insulin resistance, typical of type 2 diabetes, you end up with hyperglycemia.
High blood sugar, the hallmark of diabetes.
Exactly, diabetes mellitus.
And what about glucagon imbalances?
They can play a role too. In diabetes, glucagon levels are often inappropriately high, even when blood sugar is already elevated. This high glucagon contributes to the hyperglycemia by constantly telling the liver to pump out more glucose.
So it makes the problem worse.
It definitely can. Conversely, very low glucagon levels, though rare, could potentially contribute to problems with hypoglycemia, making it harder to raise low blood sugar.
So understanding this whole interplay is really fundamental for medicine.
Absolutely essential for diagnosing and managing diabetes and other metabolic disorders. It highlights just how critical this tight regulation is.
Wow. Okay, so let's just recap quickly. We started with the big picture control systems, homeostasis, the RCCE component.
Receptor, control center, effector. Yep.
Then the feedback loops, negative feedback, like a thermostat keeping things stable, and positive feedback amplifying things for specific events.
Right, the accelerator pedal.
We touched on circadian rhythms influencing set points. Then we dove into hormones, the PGS classification.
Peptide, amino acid derivative, glycoprotein, steroid, and how being hydrophilic or hydrophobic dictates their transport and action.
Right, the postcards versus the VIPs needing a ride, and the HPA axis as an example of hormonal control.
Central command for stress.
Then we focused on the pancreas, its dual role, and the islets of Langerhans with their alpha and beta cells.
Making glucagon and insulin.
And finally, we unpacked insulin, the storage manager, its synthesis, glucose-triggered release, tyrosine kinase receptor, and anabolic effects.
Lowering blood glucose.
And glucagon, the mobilization expert, its synthesis, GPCR pathway via gamma-P, and catabolic effects.
Raising blood glucose.
A really comprehensive journey.
We covered a lot of ground. And hopefully revisiting those questions helped solidify things. Like why positive feedback is rare for daily balance. It's just too much amplification for stable control. Useful for emergencies, not cruising speed.
Right, and the hydrophilic-hydrophobic difference really explains how hormones get around and talk to cells. It's not just random. It's based on their chemical nature.
Precisely. Form dictates function.
So here's a final thought to leave everyone with. Just take a moment and consider the sheer, unbelievable amount of information your body is processing right now. Every second. Silently, perfectly coordinating all these systems we talked about without you having to lift a finger or even think about it.
It's truly mind-boggling when you stop to appreciate it.
What deeper implications does this incredible self-regulating biological machine hold for how we think about health, about disease, and maybe even about what it means to be alive?
A profound question indeed.
Well, thank you for joining us on this deep dive today. We hope you found it useful. Keep that curiosity burning, keep asking those questions, and keep exploring the absolutely fascinating world within you.